In this screencast, we're going to talk about the fourth and final stage of cellular respiration called the Electron Transport chain, or the ETC. So as a review of the cellular respiration process itself, we've already talked about glycolysis and the Krebs cycle and the oxidation of glucose to pyruvate and how two pyruvates in the presence of oxygen would actually enter the mitochondrion and enter the Krebs cycle. At each step along the way, we're going to account for ATP molecules, and of course, the big story that we're going to cash in today are these electron carriers that we've talked about so much. So far, we've accounted for ATP molecules being made in the process of glycolysis and the Krebs cycle, and in glycolysis we had two ATP molecules that were the net result so far. And because there were two cycles of the Krebs cycle for one glucose molecule, we've got a net of two ATP molecules from the Krebs cycle. So there has to be a lot more than that. There has to be a way to harvest way more ATP molecules through this process. They are too valuable for the cells of living things. This is where the electron transport chain comes in. This is a series of proteins that are built into the inner mitochondrial membrane. And there is where these electron carriers are going to then pass off these electrons to be passed from protein to protein to protein in what's called the respiratory chain to unleash some energy. And we'll talk about how to do that. You can get about 36 ATP. Really, it's probably somewhere less than that. There's typically about 2 to 3 ATP per electron carrier that we are going to harness by oxidative phosphorylation. So remember, the double membrane of a mitochondrion is super important to its functioning. So this is that respiratory chain that we talked about. There's the inner mitochondrial membrane here, and each of these proteins set up in kind of a linear fashion there make up the respiratory chain. That's the redox carrier proteins that are embedded in the membrane. Each one is specific for either NADH or FADH2, but that doesn't matter to us. So we remember the electron carriers that have been kicked out in each of the processes so far, and we can account for them if we go back and take a look at glycolysis and the Krebs cycle. But the important part to remember is that these were kind of like money in the bank. We kept using that analogy, and we are going to withdraw all that money here in the electron transport chain. So here at the electron transport chain, or this respiratory chain that is set up in the inner mitochondrial membrane, these carriers, as you notice each of the types we've talked about, these carriers are going to be acted upon by their different substrates or their different enzymes here, and the electrons that come from the oxidation reactions are going to be passed from molecule to molecule. Now, what's also going to be donated here are going to be hydrogens, and hydrogens are going to be pumped across to set up a concentration gradient, a proton gradient. But these are indeed oxidation reactions, and if you remember oil rig, I will make mistakes myself unless I remember oil rig, that the oxidation of NADH means that they're going to be losing an electron. Okay? So here is the oxidized form as we lose an electron. Oxidation is losing. Because this is an exergonic reaction here, that's the energy that's used to power or actively transport the protons across this cell membrane. So taking the hydrogens from the electron carriers, and the electron carriers passing electrons and the protons to the electron transport, the H is going to come off, it's going to be pumped across, and the electrons are stripped from those as well, from those hydrogen atoms. So the protons are going to be pumped across actively, and the flowing electrons are going to have the energy to do work. So it's these transport proteins in the membrane that are actually going to pump across the protons, the hydrogen ions. So in the electron transport chain, the oxidation of these electron carriers is going to be an exergonic reaction, and that energy is going to be used to pump across the protons. Now, pumping the hydrogen ions, or those protons, across a membrane, it can't be ATP. So it's not the chemical energy ATP. That would cost you actually way more energy than you're actually going to make out of the process. So the flow of electrons from carrier to carrier to carrier, to protein to protein to protein in this respiratory chain, or the electron transport chain, is going to power the flow of the hydrogens, the hydrogen ions. These electrons are going to flow downhill, and eventually, at a lowest state, they are going to be accepted by the final acceptor, oxygen. So when the oxygen is going to accept the electrons from the transport chain, as well as the hydrogens that are falling back down a concentration gradient across the intermitochondrial membrane to form water. The oxidative phosphorylation occurs when the hydrogens are falling down their concentration gradient through ATP synthase, so they are going to harvest that energy to make ATP. So as a further little explanation of this concept of electrons flowing downhill, they move in steps from carrier to carrier to carrier, from carrier to carrier to carrier along the mitochondrial space there, or membrane. And this is a controlled release of energy. So you have an analogy of the electrons actually going from a higher state to a lower energy state, and what that allows us to do is allows us to harness some of that energy. It's not much different than thinking of water flowing over a dam. When you put water behind a dam, and if you release it, you can actually harvest some of that work. It was potential energy from behind the dam, but that kinetic energy can be used to harness or to be able to do some work. And we use that to synthesize ATP. So this is chemiosmosis here. We've got a concentration gradient of these protons, and when the protons flow through ATP synthase, they flow through this carrier protein from an area of higher concentration to lower concentration. This subunit of that protein has specific properties of it that we can use that energy to phosphorylize ADP back into ATP. This diffusion of the ions from one side to the next, that proton gradient is used so that we can, as they flow from a higher state to a lower state, we can harness ATP. The force of this is called proton motive force, remember, and that's simply in any substance, including hydrogen ions, but it's that tendency to spread out, to diffuse across a membrane. So the proton motive force can be kind of described as the potential energy set up by a concentration gradient. So it's a really complex idea that was thought up by this scientist here, Peter Mitchell, during the 20th century. Before that, we always thought that ATP, which we knew about, would be formed from substrate level reactions, because we knew about chemical reactions involved the rearrangements of bonds, and we would take a phosphate away from another molecule to make ATP. So oxidative phosphorylation, or chemiosmosis, or the proton motive force, was really pretty revolutionary. So here I think you'll want to stop and review these steps, just kind of saying them and showing them with a different source in mind of the electron transport, where we're going to pass electrons from a kind of a higher state of energy to a lower state of energy. And as we do that, we're able to harvest energy. So it's really easy to account for the energy that's been produced by substrate level phosphorylation so far, because that's simply rearranging bonds from one substance to another. In the electron transport, oxidative phosphorylation is not a pure accounting of ATP molecules. Of the energy or electron carriers that enter the electron transport, about 2 to 3 ATP molecules can be phosphorylized from this chemiosmosis. So even using about 2.5 as the average, somewhere between 2 or 3, we end up with around low 30s is more accurate for ATP molecules. The point is, a lot of ATP molecules are harvested from the complete oxidation of glucose and the electron transport system. As a final accounting and summary of a cellular respiration, pause here and answer these questions. Where do these substances come from? Where do they enter cells? Where do they enter or leave as the process of cellular respiration? So it's really important to note that the final electron acceptor in the ETC, electron transport chain, is indeed oxygen. So that means for aerobic respiration, a cell needs a constant supply of oxygen. And it's easy to think about what wouldn't happen in an animal cell or in a human if the oxygen is unavailable. If you're trying to run a marathon, at some point, you can't keep up with the demands for this process of oxygen. You start breathing heavier, you start to increase your heart rate to try to get oxygen pumped to all of your muscles via the blood. There's a lot of chemicals that have been discovered, one in particular called cyanide, which cyanide disrupts this process. If the electron transport backs up because there's no oxygen available, well then the electron carriers, they can't unload their electrons, and we can't have the hydrogens diffuse back across the membrane. We won't be able to produce the water here, and so the lack of oxygen really backs this up like a backed up sink in your house. Therefore, the cells are going to run out of ATP, they're going to run out of energy, and without the constant supply of oxygen, the cells are going to die. Remember, this is the point. So we should be able to go back and have a final accounting of each of the steps. What are the reactants? What are the products? And what are the energy in the form of ATP molecules that are being made?